So welcome everyone and good afternoon. So it is my great honor to introduce Dr. Lindsell. And Dr. Lindsell is the director of the Vanderbilt Institute for Clinical and Translational Research Method Program, co-director for the Center for Health Data Science and professor of Biostatistics and Biomedical Informatics at Vanderbilt University Medical Center. And his current focus is on learning health systems, leveraging data processes and data system to enhance learning from pragmatic trials and observational studies. And he has published over 300 peer-reviewed papers and has led the Data Coordinating Center for numerous multi-center clinical trials and epidemiologic studies. And he has applied his expertise in the design and execution of pragmatic effectiveness trials, risk stratifications, and predictive modeling. So I have always been jealous of Vanderbilt's infrastructure to do the pragmatic EHR-based trials. So today, Dr. Lindsell will share with us two ongoing big pragmatic platform trials on repurposing drugs in COVID-19. Okay, let's welcome Chris, Dr. Lindsell.
Thank you very much indeed for a very kind introduction. Can I just confirm that you can all hear me okay?
Sounds good.
Excellent.
Yes.
Thank you. So I appreciate the introduction. I'm going to run through today the story of Active 6, the story of Treat Now, and I'll tell a few other stories along the way. The things that we have learned about running large pragmatic effectiveness trials during COVID-19, and what we've learned about the drug repurposing programs and running decentralized trials. At any point during the talk today, if anyone has questions, please do enter them into the Q&A box. I believe it is being monitored, and I'm happy to answer those questions as we run through. So a few disclosures, very briefly. I do receive funds from NIH, CDC, DoD, and a number of other places to support the work that I'm talking about today. Particular thanks and acknowledgement go to the very large study teams that enable these things to occur. It is impossible to list the number of people, as it is well over the hundreds, that are all contributing in some major way to the ability to do the kinds of studies that I'm talking about today. So a quick reminder about the COVID context. While it seems as though we are now in the third year of a trilogy that began in 2020, 2021 was also fairly challenging, and 2022 is an interesting beginning. I just want to remind people where we were when we first started thinking about these platform trials. Back in March 2020, so it really was 2 years ago. During this period of time, mortality was increasing. There were a lot of discussion about getting trials to test drugs available, but we had no historical information about this disease, about the pandemic, and about the kinds of things that would happen. And in that setting, we didn't have an awful lot to run on. And so we just said, let's go fast. And let's try and do studies that will get really quick answers to the questions that are out there. And I'm adding a sentence here at the very bottom, because this is a sentence that I heard almost every day for a year. If we don't go faster, thousands of people will die. That is a very true statement, but it isn't the only statement, and it isn't the only thing that matters. And many other things influence the thousands of people that will die. And as we'll talk today about some of the decision-making that we have to make in this kind of a context. But in this setting, our first experience of a repurposed medication was this one. We looked at hydroxychloroquine, as many people did. 12 days it took us to go from the idea to having the funding, the grant, the protocol, the approvals, first patient in. So 12 days from having an idea to the first patient in a 34-site randomized controlled trial of a repurposed medication. In this particular situation, the biggest learning for us up front was around consent. Back at the beginning of the pandemic, again, I remind people there was not a lot of PPE to go around. Nobody understood transmissibility, how contagious the disease was, what the mortality would be like, who was truly at risk or who was not. And so in the very early days, the idea of a paper consent form or even an electronic form that somebody would have to put their hands on wasn't really okay. And we had to come up with a way to do research consent without the patient having equipment or material that they would then give back to us. We were very lucky that just prior to the pandemic, REDCap, the people at REDCap had developed a way to do e-consent, a compliant signature on a consent form that met the needs of both FDA and other regulatory bodies. And so we were able to leverage very early the e-consent process that REDCap had built out. This becomes an important aspect of our story. The e-consent system, as it is envisaged, is an easy way to inform participants about what a study is and to document their willingness to participate. Unfortunately, e-consent has also become a little bit of a challenge to us. The results of our first trial are shown here. This is the hydroxychloroquine study. On the left-hand side panel, we see the outcomes at 14 days after randomization. And we see a bar for hydroxychloroquine. And in that bar, we see at the top, the white colored, and at the bottom, the darker colored. The white colored is the proportion of patients that have been discharged and have no limitations in activity. At the bottom, the dark colors are those patients who have died. This bar reflects an ordinal variable. The ordinal variable is the COVID outcome scale. And as you can see, like many people, we measured that at 14 days after randomization, and again at 28 days after randomization. And as part of the hydroxychloroquine story, we found no evidence of benefit. Our learnings, however, were not so much about hydroxychloroquine. I call this story the tortoise and the hare because faster is not smarter. We went through this trial really, really fast, and we designed it as a Bayesian trial so that we could update our decision-making along the way. But we wrote down a frequentist analysis plan. And so we had a frequentist analysis plan and a Bayesian intent that rested in the protocol because of going too fast. In addition, there were another couple of elements of the data analysis plan that were somewhat confusing based upon how quickly we put it together. And what we discovered at the end, when we went to publish the data, the study itself was published in a reasonably high-tier journal in JAMA. We had to fight really hard. I told you it took us 12 days to go from the idea to the first patient enrolled in the trial. It took us 3 months from submitting the results to the journal to accepting the results as being valid. And the primary discussion was around the use of outcomes that were not taking advantage of the ordinality of the scale. The suggestion to use pure mortality or time to recovery were really, really strong. That pressure was intense. And that's another part of our story today, is the pressure from stakeholders to do things in very traditional ways really does change the way we think about things. And again, in this story of the tortoise and the hare, I would argue that I think the hare has lost the race and the tortoise is still going on. And we'll see a little bit of evidence of that as we go through. Very briefly, I'm going to tell a slightly different story. This is a 2-slide story, and I call this story Narcissus. Those of you who are unfamiliar, the Latin name of the daffodil is Narcissum. It's a beautiful flower, but the story of Narcissus is not so pleasant. Ultimately, in the story of Narcissus, the self-reflection, the self-aggrandizement becomes a challenge to success and furtherance. And in this particular story, building on the backs of our hydroxychloroquine study, we developed a mechanism by which DSMBs can share information between clinical trials that are currently ongoing with the sole purpose of making decision-making at the DSMB level better. The idea is that if there are 5 trials that are all studying the same drug in similar patient populations, that one trial might learn from the other trial in terms of whether you should stop for futility or efficacy at an early stage. Our findings in this particular study is that while it sounds like a really good idea, and it is technically feasible to do it, the general infrastructures that we have created in medicine and in clinical research really don't help us to do data sharing. In the absence of good, solid trust, in the absence of relationships that allow things to move forward, and in the absence of the pressure of mortality of patients, we really got a lot of pushback. And at one point, I was told by an investigator that data sharing is actually anti-promotion for academic individuals, and therefore contributing to a broader data sharing initiative went against institutional priorities. Those are some of the things that we have observed in the pandemic, and we've been able to elevate into the limelight some of the issues with the way we do clinical research. So the story of Narcissus is that by worrying too much about the self, we really don't help the whole. So back to our other story, the story of the development of platform trials in COVID. I was talking a little bit about the hydroxychloroquine trial that we call ORCID. Off the backs of ORCID and the hydroxychloroquine trial, because of our success there, we were asked by NHLBI to participate as part of CONNEX. CONNEX is NHLBI's network of networks, and their goal was to pull together all of the trials that they were sponsoring in the COVID space, and to try to improve learning by developing more of a master protocol type of approach. And as you can see from this slide, which I updated yesterday, CONNEX now has 42 networks with 6 major studies, 732 participating sites, and 65,000 participants. So certainly a lot of opportunity for learning. But this is what our charge was. Our charge was to design a trial that would get the right answer really fast. Something like I-SPY2, but in COVID. For those of you who may not be familiar with the I-SPY2 story, I-SPY is an adaptive platform trial and perhaps one of the earliest and biggest examples. And it has been running for about 15 to 20 years. And throughout its lifespan, it has been developing the mindset around what it takes to do platform trials. So they wanted I-SPY2 with a 15 to 20-year history developed in COVID in the matter of weeks. They wanted to combine drugs that target different organs, but that had one control group. They wanted it relevant for inpatients who are critically ill, as well as outpatients who are mildly asymptomatic. They want the trials that would support an emergency use authorization and new drug applications. But they also wanted the platform to be able to include prevention strategies to prevent long-term sequelae and include repurposed drugs. And so we were asked to write a master protocol that would satisfy every eventuality. And clearly, such a thing is a unicorn. And I'm not sure they really do exist. And here we see an image from The Economist of unicorns playing soccer with a coronavirus, which I think is probably the best story of 2020, as I'm not it, not my virus. But this does lead us to outcomes. As we think about the unicorn, the master protocol, we need an outcome that is relevant. And what is that outcome for outpatient platform trials? For regulatory decision-making, it's essential to protect the public and ensure that only therapies that have demonstrated benefit that outweighs the safety concerns make it forward. And so in that setting, protecting the public, preventing a false positive trial is a really important thing. Conversely, finding a small signal in a pandemic, that's really trying to find the most sensitive study design we can. We want to prevent a false negative trial. And in this situation, what we're asked to do is to try to design a trial that is like putting a square peg into a round hole. They said that it's actually not possible to do, but just because they said it can't be done, doesn't mean it can't be done. So what we set out to do was to try to design a trial that had the ability to prevent false positive outcomes and false negative outcomes. So a trial that was going to protect the public and find small signals. In this setting, use of traditional outcomes were being proposed. Things like rehospitalization or death at a singular point in time, the number of days free of hospitalization, the occurrence of things like thrombotic events. We were finding in May, June, and July of 2020 that the coagulopathies of COVID were really important to understand. And so outcome measurements that ignored the possibility of massive clotting issues, those outcomes were not considered beneficial unless you could somehow include these thrombotic events. And so we thought maybe we can do better. We started out with the World Health Organization scale. Very straightforward, just a number of levels. The worst possible outcome is death. In the middle, different levels of oxygen support that really reflect the primary focus of research and discovery on COVID-19 was around pulmonary function. And you can see at the top end of this scale, there is no clinical or virologic evidence of infection and limitation of activities is the only differentiator in that outpatient space. One of the pieces of hidden in the text of the World Health Organization scale description are these two words, over time. So measuring illness severity over time. And I think that this is one of the really important lessons for me in COVID, is that it's time for us to stop asking about questions at a single point in time and start to think about the time course of disease. So if we come back to our World Health Organization outcome scale, at the time we designed ORCID, this had 7 levels that went from death to discharged. And we measured that at a single point in time, 14 days. But what if the lives were being saved early? Or what if we were reducing the number of patients who were exposed to a mechanical ventilator? Or what if we were reducing the total number of days that people spent on a mechanical ventilator, freeing up the equipment for other patients? And so is it really the case that all that really matters is going to the hospital and dying? Or do we care about the details of this scale? And so we decided that it was important to move from measuring things at a single point in time to measuring the time course of disease. And you can see in this particular figure at the very top, while this is cartoonized, at the top on day 2, we can see that that orange bar starts to get a little bit bigger before it gets a little bit smaller again. And that's reflecting the fact that early in the disease, patients start to get worse before they get better again. And it's possible, in this case, the orange is hospitalization. Patients can go to the hospital and then leave the hospital. And so it's important to measure that. If I was only looking at day 10, I would be missing the fact that I have a lot of patients that go to hospital on days 2, 3, 4, and 5. And so we need to try to bring that into our study somehow. The statistical approaches to this were not really very good. There were some reasonable approaches back in May of 2020, but some of the issues to take on. When the outcome is an absorbing state, when a patient has died, it isn't possible for them to change state again. However, when a patient has recovered, it is possible for them to get sick again. And so not all outcome states are absorbing. And we have to find a methodology that allows people to get worse and then get better. But if they get worse a certain way, they have to stay that way. We're also asking questions about repeated measures. Do we actually need to make a measurement every single day? Or can we make a minimum number of measurements? And in some of our simulation work, we really felt that making measurements, making 5 measurements was sufficient to get the granularity of data that you need over a 15-day window. If you make 5 measurements, we really increase the efficiency of the trial. If we make 15 measurements, we're not adding an awful lot with the extra 10. And so there's a lot of methods research that we can do to understand the right time to make measurements in the clinical course of disease so that we can optimally choose when to evaluate outcomes and understand the trajectory of outcomes. Some of the things that we thought about as we were doing this work. What are the good attributes? A good outcome, it gives credit for things that matter to patients, but it should also be penalized when things that matter to doctors happen. There's no point in helping people feel better if it also makes them go to the hospital more often. We know from statistical theory that we want to have as few ties as possible and that we should have the more continuous the measure, the better. We know that the outcome should be measured over the relevant clinical time course. Early in the course of disease in COVID, people were making measurements at day 7 and day 14 because we were worried mostly about progression of disease to hospitalization and mortality. In those early studies, people were not making measurements at days 28 and below and after. And so the learning about prevention of long COVID or PASC was really minimized. And so thinking about outcomes that measure the clinically relevant time course is important. And then the other thing that was really important for us to think about was rescue therapy. During the course of a pandemic, as new treatments come along, and we find that we're able to save lives either by new treatments that prevent the disease from progressing or treatments that prevent the save a life after the disease has progressed. Either way, we want to take those things into account. So for example, in the setting of COVID, antithrombotic agents used in the hospital may actually be preventing the clotting issues that are causing some patients to die. And so that is a rescue therapy in some ways that are preventing the progression to death. And so understanding those in our outcomes is really important. So we started out just by taking the WHO scale and just adding levels. And we added a symptomatic deep vein thrombosis level. And we added that at the lower end in the outpatient end. And then we added some events that we thought were important at the sick patient end. So major events, irrecoverable outcomes. Now, this kind of a scale started to give us more granularity, and to a certain group of physicians was very palatable. But overall, we're missing a lot of things. And there are lots of questions that we were unable to answer with this. So is a severe cardiovascular issue worse or better than a moderate respiratory issue? Is a kidney issue worse than a liver issue? Symptoms and functional status are really important for outpatients, but do they matter for inpatients? And organ status, organ support matters for inpatients, but for most outpatients, they don't really have organ support. So what does matter? And what matters for families, patients, and providers? And again, early in the pandemic, people were running fast and they were not thinking about the clinical time course of disease and outcomes across the full spectrum of disease. About the middle of 2020, a very large survey was done by Evangelides et al. And really showed that patients, public health professionals, and the general public do value things somewhat differently, particularly as it relates to symptoms. And we can see from this radar plot that while it's clear, clinically important things are really valued highly by everyone, there are some symptoms that are really important to worry about. Shortness of breath is something that is of concern across the continuum. And shortness of breath is not something that ever existed in any of the scales that have been developed. So we decided that it was important to pull together some kind of a scale that mattered to outpatients, that mattered to inpatients, that properly penalized for mortality, and that took into account serious adverse events if they were to occur. And we generated a fairly intense scale. It looks a little bit like this. I am purposefully not providing it to you because this scale should never see the light of day. This was the best thinking that we could put on, best thinking we had towards the end of 2020. And what this is we took the thinking that inpatient physicians put around the SOFA score and we applied it to the outpatient setting. Now, let me unpack that for people who are not familiar with the inpatient SOFA score. The SOFA score is called the Sequential Organ Failure Assessment Score. And it is a way that every day a patient can be evaluated on multiple different organs. How bad is that organ today? What is the likelihood that organ is going to fail? Is it moving to failure? The clinicians who use it tend to like it because overall it's an average. Is my patient bad or is my patient good? However, it is not very good for a lot of more deep decision-making because it's not always certain which organ is failing and therefore which organ is driving the outcome. And when it comes to regulatory decision-making, it's more important perhaps to understand the mechanism and those things that are driving the outcome than it is just to see a change. If we go back to that competing stakeholder opinion of what is worse, a false positive trial or a false negative trial, the scale that I have on the screen here really focuses on it being worse to have a false negative trial. In other words, we are looking here for anything that points at change, worsening, getting worse, feeling worse. In the SOFA side, on the inpatient side, it's objective measurements of respiratory function, coagulation function, renal function, cardiovascular function, and central nervous system function. In the outpatient side, we use the information set from the surveys that had been done to think about the most important things that mattered to patients. It was shortness of breath, fatigue, and being able to do activities of daily life. And so we borrowed 3 scales that measure those things, and we invented a new one, and we kind of combined them together to come up with a SOFA score equivalent for the outpatient setting that says, if your fatigue is getting worse, your shortness of breath is getting worse, your symptoms are getting worse, or your ability to do your activities of daily living are getting worse, we will find it. We will see it and we will be able to act upon it. And so a very, very sensitive kind of scale. In this particular setting, we also thought we had a good analytic approach. Using a proportional odds model with an ordinal regression, we should be able to analyze these data, recognizing that everybody in the hospital looks worse than everybody who is not in the hospital. We naturally have ordering in our scales. However, we did have a few problems. We thought at this point that with a good scale and a good analytic methodology, we had really found our unicorn. Unfortunately, the kinds of feedback we were getting is that using a Bayesian framework in a pandemic may be a little bit challenging in a regulatory setting because they have to control type 1 error. Whether we are fans of Bayesian and we love the decision-making framework and the ability to learn fast or not, it doesn't actually matter if the final arbiters of decision-making have to control their type 1 error. And so we had a competition here between our preference to find things quickly and the regulatory decision, which is don't produce a false positive trial. The other pushback we got is around the proportional odds model. And this is the second time we'd heard it. In trying to publish the results of ORCID, we heard from the statistical editors that clinicians do not want odds ratios. What the clinicians want is they need to understand mortality. They need to understand hospitalization or recovery. And we kept hearing it over and over again. And so here, this was the final time we heard it. Use an outcome like mortality or recovery at a single point in time. And what we were told is that our approach, we wouldn't know what changes the outcome. It was more sensitive, it wasn't specific. We were questioned about the proportionality assumption. And it really didn't matter how much we explained the relationship between concordance probabilities and the Wilcoxon test. Everyone was quite comfortable with a Wilcoxon test, but they were not comfortable with a violation of the proportionality assumption in a proportional odds model. And everything that we were doing with all of our data was showing them that really it didn't matter. The assumptions didn't matter for what the decisions we were trying to make. But really, that we weren't going to change minds at that point in time. We really struggled with modeling state transitions, going from being healthy today to being back in the hospital tomorrow, or being on a vent today, you're not going to be at home tomorrow. And so the correlation structures were much harder to tackle. And we needed a new analytic methodology to really deal with the between-day correlations. We also needed to improve our understanding of the frequency of measurements and how to interpret the data. So with all of that, we kind of said, we birthed a unicorn, we found a unicorn, we've done something creative. Let's move into Active 6, which is the next trial I'm going to talk about, having learned about outcomes, learned about what the FDA wants, learned about what the community wants in terms of getting better faster and finding signals. Let's see if we can actually find a unicorn this time. And so we created Active 6. Active 6 was the sixth one of the big active trials that came out of the federal government. And the primary feature of Active 6 is to answer the question, what medicines improve the symptoms of COVID-19? How do you help people feel better faster? A very, very simple outset. But as you can notice in this particular cartoon, there is no mention of disease progression, hospitalization, or death. And that was very, very intentional because at this point in the pandemic, the rate of hospitalization and death for outpatients who have mild to moderate disease is under 1%. And trying to find drugs or treatments or therapies that will change that amount is going to require very, 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 very big studies. And doing those very, very big studies was really outside of the scope of what we were asked to do. But regardless, we came back to the table and we said we would like to have an ordinal outcome that includes symptoms for outpatients. We think that that's important. So from the outset, we were able to include symptomatic status as a level on our ordinal scale. What we weren't able to do at the very beginning of Active 6, though, is explain how that converted. We were measuring symptoms. How does that convert to information about treatment benefit? And this is where we resurrected our scale consideration and our ordinal regression models. And like a phoenix rising out of the ashes, we came up with a way to try to count symptoms meaningfully. We borrowed from the FDA guidance documents and we measure in each of the individual symptoms using a none, mild, moderate, or severe scale. We had intended to work with participants or work with the general public to understand how they rank each of the symptoms. And we would then be able to come up with some kind of utility-weighted evaluation of symptomatology. So we're collecting the data, we're collecting the symptoms, we have every intent to move forward and use patient utilities to guide us on how to combine the symptoms. Unfortunately, that's kind of not what we're doing. While we were able to measure each of the individual symptoms, trying to come up with agreement on which symptoms matter more and whether 2 mild symptoms add up to create a moderate symptom, or whether 1 severe symptom is worse than 10 moderate symptoms, nobody is able to really come up with any agreement. And in the absence of agreement, we're left with going back to the lowest common denominator, and we're really left with symptoms, mild symptoms, moderate symptoms, severe symptoms, hospitalization, and death. How does that relate to our outcome, though? So while we're measuring symptoms, and the FDA is very interested in this thing called time to recovery, let me ask you a question. If I am measuring recovery as the day on which you achieve your 3rd straight day without symptoms, how do you count if you die on day 2? Do you ever recover or not? What are the analytic methods we have for that kind of intercurrent event? And how do we interpret recovery in that situation? What do we do in the situation where somebody recovers for 2 days and then gets mild symptoms again, and then on the 4th day has no symptoms again? These are the kinds of questions that really make it difficult to understand a time to recovery outcome. And so we spent some time working on different kinds of outcomes called mean time unwell and days of benefit. So time to recovery, it's been used in trials. The FDA has even used it to grant an emergency use authorization. It doesn't actually count symptom burden before you recover. It doesn't handle death very well. It really, the time period within which you observe recovery is very informative to the outcome. And it's very, very difficult to analyze these data in a meaningful way. And so instead of time to recovery, we propose something called time unwell. When there is no death, then all of the time that you have is time unwell. And so when there are no deaths, time unwell is actually equal to time to recovery. But when there are deaths, you're able to explicitly handle death because you are in a sick state, you remain sick. But that still doesn't help us look at change over time. What time unwell does is it allows us to look at the average number of days of follow-up that are in an unwell state. And so you can say this time, the gray zone is the zone of time that the patient spent in an unwell state. We can shift that threshold up and down, so we can say, how much time do they spend with no symptoms? How much time do they spend with mild symptoms? How much time do they spend with moderate symptoms? But regardless of how we set it, what we've done is we've created a variable, a time variable that people can naturally understand. It is the amount of time that somebody is going to be sick for. And if we can compare that between a treatment group and a placebo group, we can say that the average number of days that you spend better treated is 1.1, for example. There are, however, a couple of challenges. It is possible that your symptom burden depends very much on how you perceive your symptoms to be. And the time unwell only looks at a single binary cut point. You are either above or below the threshold. While it's possible to compute mean time unwell for every threshold, that's a lot of computation and is a little bit difficult to interpret and say, well, for mild symptoms, your mean time unwell is 7. For severe symptoms, your mean time unwell is 9. Trying to explain that to a patient is really difficult. And so rather than looking at the time unwell over time, we're looking at something called days of benefit. And if I say the days of benefit in words, really on any given day, a patient may experience, if you are in group, may find that if you're in group A, the outcome is better. If you're in group B, the outcome is better, or the outcome is the same whether you're in group A or group B. The number of days of benefit is the average number of days for which being in group A gives you a better outcome, or the average number of days where being in group B gives you a better outcome. It really is a simple translation of probabilities. And so here, even if you have a very different set of symptom profile outcomes, the difference in the days of benefit is robust. It is essentially the number of days where being in the treatment arm gives you more people in the higher levels than when you're in the non-treatment arm. And so in a situation where symptom severity is different, you can have a benefit in terms of the treatment. Even if the amount of time you spend unwell is the same, you may feel better during that period of time. And for those of you who may have had COVID or a breakthrough case and have experienced the fatigue and the cough and the dyspnea, a shorter period of time isn't the only thing. Just reducing those symptoms would be highly beneficial. And so we've moved from a single point in time with a single measurement of time to recovery or time to death to this situation where we are measuring symptoms, we are measuring symptom severity, and we're measuring it over time. And we're now using a model-based estimand, the days of benefit, to describe the outcome. Unfortunately, as we got to the end point here, and this is the end of my unicorn tale, this particular work of art is called the Hunting the Unicorn, or the Hunt of the Unicorn. And unfortunately, our unicorn was somewhat destroyed at the last go around, because while we think we have still found an outcome that is very sensitive, that satisfies all of the requirements that were laid out by regulatory decision makers, and that really does weave between the patient-centeredness of symptoms and the need to respect hospitalization and death, people really still were not comfortable with what we'd done. And so we actually did a trick. And the trick that we've done is we have allowed our unicorn, or we've allowed our unicorn to become the phoenix that rises from the ashes. And we are now using it as a screening outcome in Active 6. And so Active 6 at this point has something in it that I believe is unique. I have not seen it done before. It came out of a meeting with the FDA in which we discussed the relative benefits of a sensitive outcome versus a very specific outcome that would prevent a false positive trial moving drugs into the public domain. As we talked about this, it became very clear that Active 6 was a little bit like a seamless phase 2, phase 3 trial, where in phase 2, we're really interested in finding any signal. But if we see any signal, the regulators would like us to take another step and look at the specific outcomes of hospitalization, death, and time to recovery in the way that they would like it measured. And so to this end, what we currently have incorporated in the design of Active 6 is a highly sensitive, highly innovative screening outcome that uses a very, very unique, newly developed statistical approach that should allow us, with just a few hundred patients in the trial, to say there is possibly something here, or there is absolutely nothing here. Once we've answered the question of whether or not there is something or nothing, if there is something, we move straight into the main, the primary analytic environment for the trial, which is we move out of that phase 2 screening space into that phase 3 regulatory decision-making, and we change the outcome from being this very sensitive multi-level outcome measured every day to being a very specific outcome of hospitalization or death or time to recovery. There is something very subtle in here. It is 2 possible outcomes. The timing of the choice of outcome is actually right at the point of doing the interim analysis. We do not believe we know enough about COVID-19, about the evolving event rates, about the evolving symptomatology in the presence of new therapies and vaccination to be able to pick one over the other. If we really care about saving lives, and we all do, we need a big trial to demonstrate a benefit on hospitalization or death. The likelihood of finding that difference is very, very small. And so we've designed a trial that says, we will look for it. If it isn't there, that's okay. We'll look for something different. But if it is there, we still have the framework and the platform against which we can chase that outcome. And so this was a way of having our cake and eating it as well. If we find that the event rates are too low to have any meaningful outcome in the trial, then we pivot and use time to recovery as our primary outcome, using recovery as described by the FDA of a certain number of days free of symptoms directly assessed. And so we've moved from clinical event, or we have this scenario of having clinical events and symptom burden. And I'm going to pull this talk to a close and open it up for questions by moving forward with an observation. The observation that I have is that throughout Active 6, we've talked about clinical events and we've talked about symptoms. We've invented outcomes that allow us to include symptoms. The whole purpose of the trial was to make patients feel better faster. And so the rigmarole that we've gone through to try to satisfy all stakeholders makes us ask the question, are differences in symptom burden meaningful? Or more importantly, are differences in symptom burden meaningful in a regulatory setting? And I would posit that at the end of the day, it isn't really a statistical question. It's more of a question of setting priorities and trying to understand what hurdle we're trying to cross, what bar we're trying to cross, what is the right bar for decision-making. And one might argue in the setting of repurposed medications for something like COVID-19, where we are looking at drugs that are very safe, they are on the market, they're available, they're known. What we really want to understand is, are they helpful for COVID-19? In that case, is the right bar the same bar as it would be for a brand new de novo molecule for requesting an emergency use authorization? Or can we use evolving knowledge to build up towards that bar for an FDA submission? Or are we always going to be stuck taking lots of small steps unless we consider the big regulatory bar at the end of the day that we have to overcome? Ultimately, if we do question regulatory decision-making, I will share the story of Marie Antoinette. Ultimately, she was beheaded, not because she was the bad decision-maker, but because the general public decided to question the decision-making of the leadership of the time. So I will ask the question, what is the right bar? But I will tell us that there are major consequences of if we change it too much, too quickly, or without careful thought. And so with that, I will finish my talk today and say thank you very much. If anybody is feeling symptoms of Active 6, feel free to sign up. We are currently accepting volunteers. And to give you a sense, we are enrolling 400 patients a week in this trial. So thank you very much, Lady. Back to you.
Yeah, thank you. Thank you. It's really a nice talk. I really enjoyed it. So let me ask first the audience, does anyone have any questions? I actually have a question because I was kind of shocked when you mentioned that only took 12 days, when the JAMA paper published until you enroll the first patients. So we know, the whole process take a lot of process, like how to prepare the protocol database, IRB, and how you made that happen, like 12 days. Can you just give us like something more detail and just how you can make that work?
I absolutely can. I am just going to share my screen again. Okay. So I can talk very quickly about how we did this. Okay. So this is the design of Treat Now. Treat Now was the platform that preceded Active 6. With Treat Now, the entire trial has to be completed without ever having any physical interaction with a participant. The way that we can get the timeline from this kind of a statement to a database is by this kind of work. I sat my research data people, my statisticians, and my clinicians in a room, and I made them go through all of the decision-making. What forms need to be filled out by what patient at what time to get the right data in the right place. This is the only way that I know how to do it, is you get everybody together, you map the trial, and then we understand the cross-section between the data generation, the data collection, and the data analysis.
I see. So you have good people and good infrastructure to support all this, like when you have the idea until implementation, right? So you have really a nice infrastructure and people to support it. I mean, there's no way for our group to do it like in 12 days.
So I will tell you, Lily, when we started ORCID, it was me and my data manager. It was two of us. And we built it with an application developer who was able to help us with the custom coding outside of REDCap. So we did the whole thing ourselves.
Oh, wow.
And so it is possible to do it. We were very lucky because those words, if you don't go fast, people will die.
Mhm.
The IRB, the FDA, all of our colleagues said, we will help you.
I see.
Because people said they will help, you can do it.
Okay. Yeah, it is amazing. So can I ask, maybe I can ask another question if no other questions. So because you mentioned your outcome is multi-level outcomes and also it's ordinal data. Is it possible, you can give like decision theory, you give a cost, like utility, right, or cost for each outcome, then, for example, the death is the most severe one, give a high cost, then sum over this weighted, kind of weight the outcome and do like weighted score. And using
I love that. I think I absolutely believe that we should do that. The challenge is to understand the true cost. And is it a financial cost, a societal cost, a personal cost?
Mm-hmm.
And that is very hard. But I really believe that as we understand outcomes, I get why the FDA has to make the decisions they do. I understand the need for a simple outcome. But I think that while we understand the need for regulatory decisions,
Mm-hmm.
for non-regulatory decisions, there is so much information that we ignore when we don't take into account the levels in an ordinal scale, and we don't consider how or why something might be more important. A good example is stroke. If you're familiar with stroke, they use the modified Rankin scale as the outcome. The utility-weighted modified Rankin scale gives a different value to death than the main scale. In the main scale, death is 6, and if you are disabled and you need somebody else to look after you for the rest of your life, you're a 5. The utility-weighted switch, and patients prefer to be dead than to be in a state where they are not competent.
I see. Thank you. So we have 2 questions in the chat. The first one is, the question is, when you say time to recovery, how do you differentiate active virus versus long COVID symptoms?
Yeah, that's a really, really good question. What our data are showing is that in the mild to moderate sickness, most movement in symptoms occurs within the first 10 to 14 days. And so during that period of time, we see a very dynamic change in symptoms. After about 14 days, there is very little left in the dynamic nature of the symptom. They have plateaued. And so what we are looking at, our estimator, I don't believe you can measure time to recovery by the number of days that somebody is feeling good, for the very reason that the person who asked the question asked it. Not everybody recovers. And so because of that, it's important to not assume everybody recovers. Another approach has been put forward, a frailty model could work here, or a cure model, where patients who develop PASC are actually more frail, or they're cured, if you will.
Okay, thank you. So another question related to the multi-level outcome. So Kathy is asking, can you consider the win ratio approach as an endpoint?
Yeah, we actually like the win. I like the win ratio a lot. The problem with the win ratio is it has validity inside the trial and it has meaningfulness inside the trial. But try to explain to a patient what the clinical benefit of the treatment is on the basis of a win ratio. And so that was actually one of the reasons why I like the mean time unwell or the day's benefit, because it provides something that a patient can understand. with a win ratio, it's a really sensitive outcome, but you're comparing patients inside the trial to other patients inside the trial. You're not externally valid, if that makes sense.
I see. Kathy, did, okay. Kathy is really, really true. Thank you. Another question is, did you have the authority to define the decision criteria using the people with relevant expertise?
Oh,
authority.
This is a big question. What I have discovered in my long career is I actually have no authority for anything, but it's always my fault. The real answer to your question, though, is in terms of the decision-making framework, we have been very influential in the decision-making, but in clinical trials, ultimately the decisions that a DSMB makes, nobody understands them. We can give a DSMB very fixed decision rules, and they can come back and say, don't do that, do something different. And so the authority for the decision-making in a trial is really the Data and Safety Monitoring Board.
Okay, thank you. Is there any other question I missed, James? I think that's all the question in the Q&A and chat. Okay.
Correct, that's all I'm seeing as well.
Okay, can I ask a final question? So when you run the trial, there's a complexity of like patient, like for example, was assigned one treatment, but later it didn't work, right? So you switch to another treatment in the middle. So when you talk about the multi-data longitudinal, it looks like you have a setup window like 14 days or 30 days. But what if some patient like switch to another treatment or drop out, and how you're going to handle this? And it is maybe common, right?
Yeah, it's a good question. And one of the things that I think about now is that none of us ever experience any kind of illness and have no intercurrent treatment. Even if all we do is we go to bed early that night, we're all doing things to make ourselves feel better. If we model the data longitudinally and we consider each day's covariates, and we think about modeling the state transition as a function of today, then we can start to take into account things like this patient started a new kind of concomitant medication. So I think it's important for us to consider time in everything we do and the journey of somebody's symptoms, outcomes, or signs.
So dynamically making the decision based on updated patient information. I see. Okay.
Yeah.
Okay. Thank you. Okay.
It makes it harder to do the analysis, and it makes it harder to collect the data. But in truth, people have longitudinal lives.
And also it's more useful to the patient, right? Because it can dynamically tell you what's the next step rather than wait until the very end. Yeah.
Oh, yeah.
Yeah. Thank you so much. I think this is very interesting. And thank you, Dr. Lindsell, again, and thank you all for coming to this Zoom seminar. Okay, thank you. Bye-bye.